Okay, in this lesson we'll take a look at the role of the nucleus in reproduction and heredity. So we've just learned from Spallanzani's work and then later people in the mid-1800s that animals reproduce by producing sex cells. Although Spallanzani got it wrong, he thought the fluid had fertilizing power. We now know it is the male sperm cell that has the fertilizing power, and of course females produce eggs. And it's the union of the sperm and the egg that produces the next offspring, right? Well, the question then was, well, what is it about the sperm cell and the egg cell that has this information to make a new individual? In other words, where in the cell is the hereditary material located? Now, in the mid-1800s, biologists were proposing what we'll call the cell theory, that all living things are composed of cells. And when researchers examined cells under the microscope from many different kinds of organisms, they did find a structure that was present within the cell called the nucleus. Sex cells also had a nucleus, so sperm cells and egg cells had a nucleus. So it raised the question, perhaps the nucleus might be a hereditary structure. And so some early researchers in the late 1800s did some simple experiments to start to investigate the role of the nucleus in cell activities. Here's a picture of an amoeba. Here's a food vacuole with a paramecium, and this structure over here is a nucleus. Now, in some species of amoeba, there are multiple nuclei in the amoeba. In other species of amoeba, there's one nucleus, like we find in our body cells. Let's take a look at this amoeba. Let's say here's the nucleus of the amoeba. Some of the early researchers, what they wanted to do was to test the role of the nucleus in survival and reproduction by cutting the amoeba in half. So if you cut the amoeba here, sometimes the membrane will reseal, sometimes it'll kill the amoeba, but sometimes you'll get lucky, the membrane will reseal, and that'll form one part of an amoeba here that has the nucleus, and then another part of the amoeba full of cytoplasm, but it has no nucleus. And so the experiment was to simply observe what happens to the two parts of the amoeba, one that has a nucleus, one that doesn't. We can use our thinking format. If the nucleus is necessary for survival and reproduction, and a large single-cell amoeba is cut into two parts, one with the nucleus, one without, the prediction that follows then is the part with the nucleus will be able to survive and reproduce. The part without the nucleus will not. The results are the same as predicted. That's what you find, or that's what they found. And so our interpretation is that the hypothesis is supported, that the nucleus does or is necessary for survival and reproduction. Now, in the early 1900s, a researcher by the name of Hammerling did a series of experiments on a green alga. This is a single-celled green algae, right? And it's called Acetabularia or Acetabularia, different pronunciations. So Acetabularia is pictured down here. It is one cell, but it can be a centimeter or two high, so it's easily visible with the naked eye. And it has three parts to the cell. So the foot part is where it's anchored, right? And that's where the nucleus is. Then the stalk, and then the cap. So three parts to this organism. And Hammerling then is going to do a series of experiments to investigate the role of the nucleus in the life of this single-celled organism. It is an algae, so it's photosynthetic. It does photosynthesis. But he was interested in the role of the nucleus, not in photosynthesis. So what does he do? Well, he discovers something interesting about Acetabularia, namely that it can regenerate. In particular, if you cut off the cap of the green algae, you'll find that the foot piece can regenerate a new cap. If you think about regeneration, it's sort of similar to reproduction because the cell has to have information and building materials to make the missing part. So just as animals, the males and the females, must have information to make offspring that they pass on in their sex cells, so too a single cell, when it regenerates, has to have information and building materials to make the missing part of the cell. So when you cut off the cap, Hammerling found that the stalk piece will regenerate a cap. So there must be building materials and information to do so, located somewhere else in the cell. Hammerling was interested whether that location was the nucleus, where this information might be residing. So here's a diagram of this single cut experiment where he's going to cut off the cap, the foot piece regenerates a cap, and now let's think about what we would predict to happen to the cap piece itself. Okay, so here's our reasoning. If the nucleus controls regeneration, which is a form of heredity, and the cap of the unicellular green alga is cut off, then the cap should or should not be able to regenerate a foot. Well, since the cap does not have a nucleus, it should not be able to regenerate. So it turns out the results are the same as predicted. The cap does not regenerate anything. Interpretation would be the hypothesis is supported. Again, so the foot piece that has the nucleus can regenerate. The cap piece does not regenerate anything. It does not have a nucleus. It simply dies. Now, Hammerling went on to do another experiment, again testing the hypothesis that the nucleus controls regeneration. But this time, he doesn't make one cut, he makes two cuts. And when you make two cuts, you have something like this. You have a foot piece with a nucleus, you have a stalk piece, and then the cap piece. Well, if we make our predictions, remember just like the single cut experiment, the piece with the nucleus we would predict will regenerate. And with these other two pieces, since they don't have a nucleus, we would predict that they will not regenerate anything. Right? Now, we would put down here for our prediction then that the foot piece will regenerate, the other pieces will not. The results turned out to be interesting. These are the results. The foot piece did regenerate, but the stalk piece also regenerated a cap. And the cap dies just like the other experiment. So this is a puzzle here, this one here, because after all, there's no nucleus in this stalk piece. Yet the stalk retains the power somehow to regenerate a cap. And that was interesting to Hammerling. We would think about how might you interpret these results, right? So what kind of hypothesis could we come up with to explain why it is that the stalk piece regenerates a cap? So think about that for a moment. Well, one possibility is perhaps the nucleus produces some kind of substance that diffuses up into the stalk. And as it diffuses into the stalk, right, it would reside in the stalk so that even if you remove the stalk, that substance will still be in the stalk, and that substance has the power for regeneration. So the reason the stalk has the power to regenerate is because it was formerly attached to the foot, and the nucleus was producing a material that diffused up into the stalk. So in this hypothesis then, the nucleus is critical, right? It's producing a substance that has the power for regeneration. Now we can think about ways that we might test this idea that the nucleus is producing some such substance. So take a moment to think about that. Now some possible ways to test this idea, maybe somehow you could block the material coming up from the nucleus, right? And then the stalk would no longer have its power to regenerate. Hammerling did something different. What he did was he cut off the first regenerated cap. So here's the stalk. It doesn't have a nucleus. It has regenerated once, and then what he did was he cut that cap off again, and he found that there was no second regeneration. And that would support the idea that perhaps these nuclear molecules here, maybe they get used up, for example, in building the cap. And so once they are used to build the first cap, there's nothing left to build a second cap. That would explain why there was no second regeneration. So one possibility for what these nuclear molecules are maybe are building materials. Perhaps they are produced by the nucleus and they are used to build the cap. And once they're used to build the cap, they can no longer be used to build a second cap. But that's not the only possibility for what these molecules are. In this slide here we see a second possible hypothesis. Here's the first one that we just said, that maybe the nuclear molecules are building materials and they get used up in regenerating the first cap. But another possibility presented itself is maybe these nuclear molecules are information molecules. In other words, they're not building materials, but they have the information to build a cap. Yet these information molecules have a short lifespan, so to speak. They're not alive, but they have a half-life, so they degrade over time, right? And so they're not around to regenerate a second cap. So we have two possible hypotheses, that the nuclear substance here, they are building materials or they are information molecules. Here are two possibilities. If they were building materials, perhaps they're like proteins. Maybe the nucleus is producing proteins, and these are the actual protein building materials to build the cap. Or perhaps the nucleus is producing some molecules that are not themselves building materials, but these information molecules are somehow used to build proteins, which are the building materials to build the cap, right? So again, the two possibilities, building materials or information molecules. And for why it is that you only get one regeneration when there's no nucleus, well, if they're building materials, they get used up in the first cap. If they're information molecules, they degrade over time. So what we need is a way to test between our rival hypotheses. Now to test, so consider this possibility. To test the rival hypotheses, the student proposes to grow the algae using radioactive nutrients. By doing so, all molecules assembled in the nucleus will be radioactive, and so detectable by radiation detectors. In effect, the nuclear molecules will be tagged in a way that allows the researcher to trace where the molecules go. Will this be a useful experiment to test the hypotheses? Why or why not? So again, here's the idea. To figure out the identity of these nuclear molecules, are they building materials or information molecules, a student proposes that you tag them with radioactivity. How do you do that? Well, you sort of supply nutrients to the algae that are radioactive, and so whatever is constructed, whatever molecules are built in the nucleus will now be radioactive, and then you can detect radiation from those molecules with radiation detectors. So wherever these molecules go in the cell, you'll be able to detect which part of the cell has those molecules, which part of the cell is radioactive and therefore has those nuclear molecules. Now, ask yourself then, to figure out if this is a good experiment, ask yourself whether the hypotheses make different predictions. So we want to think about where the radioactivity should be located if the nuclear molecules are building materials or they are information molecules. Right? And so if this is going to be a good experiment, the two hypotheses should have different predictions about where radioactivity will end up. Will it remain in the stalk, or will it end up in the cap? Right. So under the building molecule hypothesis, that hypothesis predicts that radioactivity will be located in the cap. Those building materials are used to build the cap. So if you make the nuclear molecules radioactive and the nuclear molecules are building materials, then the cap will be radioactive. But down here, the information molecule hypothesis predicts that radioactivity will be located in the stalk, because those nuclear molecules are not actually used to build the cap. They just hang out in the stalk as information molecules that are used to actually build the building materials, which then go up into the cap. Visually, we can see it this way. For the building material hypothesis, the nuclear materials are building materials, and so if we make them radioactive, the cap will become radioactive. If the nuclear materials are information molecules, the stalk will remain radioactive, and then the new molecules, the new proteins that are built in the cytoplasm up here in the stalk, they will not be radioactive, and so the cap will not be radioactive. So notice we have two different predictions, depending on which hypothesis is correct. And that's a good experiment. The results turned out that the regenerated cap is not radioactive. Right? The cytoplasm is, so the stalk is radioactive. So we can ask which hypothesis supported, which is weakened. Right? Well, that's going to support the information molecule hypothesis. Right? So the cap was not radioactive, rather the stalk was. So that supports the idea that the nuclear molecules are not building materials, they're information molecules. So at this point then, our tentative conclusions here that the nucleus releases molecules that carry information to regenerate a cap. These molecules are not actual building materials, rather they have the information to build cell parts, like the cap. And since cell parts are proteins, this information is the information to make proteins. Okay, so that's sort of where we are at this stage. Now, Hammerling did not do these radioactivity experiments. These were later experiments carried out after Hammerling's work. So everything we just learned here about the radiation here, that was not done by Hammerling. However, Hammerling did do the next experiment, and that's a transplantation experiment.